Okay, so now we are ready to talk about fat digestion and absorption, which is a little bit more complicated than the digestion and absorption of carbohydrates and proteins. Um, this occurs with the help of a substance called bile. So I want to tell you a little bit about bile. Bile is a substance that is mostly water, but it also contains a variety of components with a variety of functions. Okay, so we have bile salts, which is kind of the main thing that we're going to be talking about here. They help to digest fats. Okay, bile also has bile pigments in it. Those are largely breakdown products from hemoglobin. Bile also contains cholesterol. It also contains toxins cleared from the blood. Bile is made in the liver. So liver is shown here and also here. Okay. And then it's stored in the gallbladder. So the gallbladder is shown here in green. Okay. And then during meals, what happens is that bile, basically the gallbladder contracts to squirt bile into the duodenum. Yeah. Okay. All right, so now we're ready to talk about fat digestion and absorption. Okay, so first of all, I want to start by saying that fats are going to enter the small intestine in the form of large droplets. Okay, so remember that fats are hydrophobic. They're not going to want to just kind of float around freely in the hydrophilic space that is here in the small intestine. So they're going to kind of huddle together in these droplets. Okay. Next, what happens is we have bile salts that come in and they help to break up those large fat droplets into smaller fat droplets and turn them into an emulsion. Okay, so let's take a closer look at bile salts, what they are like, what they do, how they do this job in this slide right here. Okay, so bile salts, again, just a reminder, they're made by the liver and they help break up fat droplets. They're stored in the gallbladder. Um, they are what we call amphipathic. I think you've probably heard this term before in other classes. Amphipathic means that one side of the molecule is hydrophobic and the other side of the molecule is hydrophilic. Okay, so that is what we are seeing here, this kind of in this green blob. This is meant to depict a bile salt. So this part is supposed to be the hydrophobic side, and then over here is the hydrophilic side. Okay. And the way that the bile salt works is that the hydrophilic side will kind of face, well, let's start with the hydrophobic side. What will happen is the hydrophobic side will face the fat droplet, okay, because the hydrophobic side is going to like to be with other hydrophobic molecules, okay? And then the outside of the bile salt is going to face the aqueous environment of the small intestine lumen. Okay, so it's going to associate with the water there. So our lipid droplets are going to end up looking like this with the hydrophobic side of the bile salt here and the hydrophilic side out here. And that is going to help our larger fat droplets exist as smaller fat droplets. Okay, or it's going to help them be more comfortable, so to speak, um, breaking up into those smaller fat droplets. Okay, so moving on, remember that we had our big fat droplets. We turned them into an emulsion of smaller fat droplets with the help of our bile salts. What's going to happen next is we are going to have a breakdown of the actual fat molecules. So chemical digestion of the fat molecules into smaller component molecules. So we're going to break them up into monoglycerides and fatty acids. And then we're going to store them in these even smaller droplet-like structures called micelles. Okay, let's take a closer look at that process right here. Oops, what happened? Oh, okay. All right, so taking a closer look at this process, first of all, I want to start by saying that most of the fats that we ingest, about 90%, are triglycerides. Okay, so the chemical structure of a triglyceride is shown here in cartoon form. Okay, basically, a triglyceride is going to have a glycerol backbone that's shown here as this yellow rectangle. And then it's going to have three fatty acid tails. So the fatty acid tails are these brown zigzags that are coming off of the glycerol backbone. Okay. What lipase does is it is going to break down the triglyceride into a monoglyceride, right? So a monoglyceride is going to be the glycerol backbone plus one fatty acid tail. So it's going to break it down to into a monoglyceride and two free fatty acid tails. Okay. So that's what lipase does. Lipase is an enzyme that's made in the pancreas and is present in the small intestine. And it's going to do that breakdown. We also have some assistance from a molecule, a protein cofactor called colipase. And what colipase does is it displaces the bile salts in order to allow lipase to access the triglycerides. So that's the role of lipase and colipase. Um, Once we have our broken down products here, they are going to be stored in these structures called micelles. So this is kind of a diagram of a micelle. You can see that it's got some monoglycerides, free fatty acids. I also want to point out that other hydrophobic molecules, sorry, are going to be stored in the micelles. So for instance, cholesterol, also like fat soluble vitamins and things like that. Okay. All right, back to this process of fat digestion and absorption. Again, I want to remind you, we had our large fat droplets. We made them into smaller fat droplets with the help of bile salts. And then we took the triglycerides in there, broke them down into monoglycerides and fatty acids, and stored the products of that in these even smaller fat droplet type things called micelles. Okay. All right, so from the micelles, what's going to happen is that the individual fatty acids and monoglycerides are going to just diffuse across the membrane of the epithelial cell in the small intestine. Okay. I also just kind of want to mention on the side, cholesterol is transported using a transporter. I'm not really focusing on cholesterol here, but it's part of the figure, so I need to talk about it. Okay. All right. Once we have our monoglycerides and free fatty acids in the cell, they enter the smooth ER, where they then get resynthesized into triglycerides. Okay. Once we have triglycerides resynthesized, they combine with cholesterol and some proteins to make these structures called chylomicrons. Okay. The chylomicrons are then packaged in the Golgi apparatus into vesicles. Once those vesicles form, they are exocytosed. So we end up exocytosing our chylomicron. And then we are almost ready to reabsorb or to absorb this chylomicron into our bloodstream. But it's not as easy as just having the chylomicron directly enter the bloodstream. The chylomicron cannot directly enter the bloodstream because it is too big to fit across the capillary wall. Okay, so what happens instead is that it enters the lymph system via these dead end structures called lacteals. Okay, so they are able to go in there. And then I want to remind you that the lymph system is going to drain into the vena cava. And in that way, the chylomicron is then going to be able to re-enter the bloodstream or enter the bloodstream. I guess it wasn't there to begin with.